Today we'll take up another new topic in Unit 5, tumor immunology. There is a lot of work going on in this field, yet there is no proper cure found for cancer till date. Many Nobel Prizes have been won by the people in the cancer biology, but still there is no cure for cancer. Human body, when the cancer occurs, develops the immune response against cancer because cancer cells also express antigens. And you know, cancer is the abnormal growth of cells. And these cells are derived from a particular tissue. There are two types of cancers. And cancer is also called as tumor. Tumor means abnormal growth of cells. The tumor can be spelled out as T-U-M-O-U-R or T-U-M-O-R. Both ways it is correct. A tumor, that is an abnormal growth of cells. Now, abnormal growth of cells that takes place in the body need not be always a solid tumor. A solid tumor is the one which is derived from the tissues that are present as solid tissues. Like, say, for example, tumor or lung cancer from lung tissue, oral cancer from the oral tissue, tongue cancer or esophageal cancer, gastric cancer, intestinal cancer, hepatic cancer, pancreatic cancer, like that. All these are from the solid tissue and they are called as solid tumors. And you have heard about the blood cancers. The blood cancers could be of the T cell origin. They are called as T cell lymphomas, or they could be B cell origin. They are called as B cell lymphomas, or they could be originating from the myeloblasts, and myeloblasts are the pre-immune cells which give rise to all sorts of granulocytes. Pre-myelocytes are supposed to be to all sorts of granulocytes like neutrophils, eosinophils, mast cells, etc. And if the cancer originates from the myeloblasts, it is called as myeloma. Okay, myeloma, which is another type of cancer. So different types of, and those cancers which are of the blood origin, that is from the hematopoietic tissue, are called as non-solid tumors. Even you use the word non-solid tumors or non-solid cancer. Here the cells will be in large numbers. They will be premature. They will not be performing immune function, circulating in the body, and when you do the differential count of the cells in the blood smear, you'll see the population of these cells in abnormal number. That is the indication that it is a non-solid tumor. So, solid tumors or non-solid tumors, irrespective of the type of tumor, they express strange antigens on their surface. And these strange antigens are generally not seen in normal cells. But if at all they are seen in normal cells, in the cancer cells, these strange antigens are overexpressed sometimes. So based on the type of antigens that are expressed by the abnormal cells, or cancer cells are also called as transformed cells. They have undergone transformation. There is some kind of genetic transformation in them, and that genetic transformation is brought about by the genes that have gone haywire. The gene expression has gone haywire. Some mutation might have taken place, and that has caused this abnormality, which has given immortality to those tumor cells. They are growing beyond proportion. They are immortal. They don't die. They don't show apoptosis. and they don't behave normal and nothing works for them. Because of that, the tumors are said to be immortal cells. Now, why they are called as immortal cells? When you transplant a tumor from a person who has developed the cancer to a healthy individual, which is not permitted. If you take the animal examples, a mice has developed tumor, let us say skin cancer, mice, and you remove the tumor from the mice, from the skin, and transplant it into another mice. And without matching MHC, if you transplant, also the tumor will be accepted by that mice. And the mice will grow the tumor on its skin. In normal circumstance, when you transplant the skin from one mice to another mice without matching the MHC, that skin is going to be rejected. And we have done that in transplantation immunology. Easily, you can't transplant the organs or tissues. They are going to be rejected. But one special feature about the cancer is that it can be easily transferred to the other individual. Therefore, one need to be very careful in blood transfusion also. Without knowing if somebody has begun leukemia and he has not undergone test and he donates the blood and those leukemic cells get transferred to the another individual without the knowledge of the person who has given the blood, possibility is that the other person who has received the blood also may develop leukemia. Such dangers are there with cancer. So tumor tissue will be easily accepted. It can be transplanted. It is transplantable. And based on that, tumor antigens are classified into two different categories. The categories are: one, tumor specific transplantation antigen, that is TSTA in short form, tumor specific transplantation antigens. The word transplantation is used here because I've already told you that tumor can be easily transplanted. And after that, that rejection of the tumor by the recipient depends on how the immune response of that recipient behaves. Whether the tumor suppresses the immune response in the recipient or the immune response of the recipient will be stronger and rejects the tumor. It depends on both. Then the second category is tumor associated transplantation antigens. These are two different types of antigens. I will be dealing with their details separately. Let us briefly mention about tumor specific transplantation antigen. Then I will classify them into 6 different categories. Tumor specific transplantation antigens can be classified into 6 different categories. And they'll be unique to the tumor. Why? Because they have occurred or they have formed in the tumor due to a mutant gene expression. Suddenly or spontaneously, some mutation might have taken place. And that mutation has resulted in producing a new type of protein, which is not generally seen in normal cell. And that new protein is processed by the cell and expressed on class 1 MHC as an antigen, or it can be expressed directly on the membrane of the tumor cells. Okay, membrane of the tumor cells as some glycoprotein or as protein, as a special receptor to facilitate the cancer growth. So such strange proteins, which are not generally seen in normal cells, but suddenly make their appearance in the tumor cells, give the tumor cell that abnormal ability of growth. And since these antigens are processed in class 1 MHC and presented on class 1 MHC, you know, when the antigen is presented on class 1 MHC, it is going to be recognized by the cytotoxic T cell, which results in CTL response, and NK cells, which are there policing your body. Despite this response, like initially when the cancer occurs in an individual, there will be tremendous amount of immune response against the cancer cells, and large number of cancer cells die because of the CTL response as well as NK cell. And you know that when the NK cell and CTL response takes place, that area becomes highly inflamed because both are known to be capable of releasing inflammatory cytokines. Therefore, a tumor or a cancer, solid tumor or a cancer, is considered to be an inflamed tissue area. It shows all the inflammatory symptoms. Okay, and these unique tumor antigens, which initiate the immune response in the beginning, will succeed later. These tumors will evolve in such a way that they will succeed later to suppress the immune response. And how do they suppress the immune response? And how do they evade the immune response? We'll be talking about that in the later part of the lectures, coming lectures. But today, we'll talk about only the antigens. Despite all this mechanism, tumor antigens are expressed, there is immune response, yet the tumor succeeds in killing a person. If the proper treatment is not done, there is a possibility that the person's life will be cut short. And that is the capacity of the tumors, which can suppress as well as evade the immune response. Then the second category of the antigen, I will be talking about details of tumor specific transplantation antigen little later. I will tell you about the tumor associated transplantation antigens. These are the antigens which are not unique to tumor cells, but overexpressed in the tumor cells. Means excessive expression takes place. Generally, these antigens are expressed in the embryonic condition in all individuals, and they are necessary. They perform necessary functions in the embryonic condition during the development of the embryo. There could be even differentiation antigens and playing a very important role for the differentiation process of the germ layers and giving rise to different types of tissues, and some of them will be specific to particular tissues. All these antigens are called as fetal antigens. Tumor associated transplantation antigen. Please differentiate between the two, tumor specific and tumor associated. Both are different. And tumor associated antigens express in the fetal condition, in the embryonic condition, necessary for normal cell to undergo the process of development, the fetal development. And the embryogenesis takes place, embryo is formed, and then finally the embryo will grow and the embryo is born as a fetus. And that process of differentiation and all is a normal process. Now you imagine in the adult individual, these embryonic antigens, majority of them are shut off. They are switched off because there is no need for the expression of embryonic antigens in the adult tissue. And only the genes that are responsible for the performance of the normal function of the tissue, because it is a differentiated tissue performing a normal function of that particular tissue, those genes will be kept active. Majority of embryonic genes will be shut off. For strange reason, in cancer cells, these embryonic genes are switched on. And it is always said that a tumor is a de-differentiated cell mass. Please note the terminology. De-differentiated means it has undergone to the embryonic condition. And you know, in the embryonic condition, the cells have higher capacity or their proliferation rate is much higher because they have to grow. And the fetus has to grow. Therefore, in all the embryonic conditions, the rate of cell division is much higher. They are, in fact, non-differentiated cells forming the differentiated cells, that is tissue. And in the differentiated cells also there is growth up to a particular stage. After that, growth stops and the cell cycle is inhibited. There is regulation of cell cycle. Only when there is death of cells due to some mechanical injury or due to some toxic injury, the tissue tries to replace the dead cell by dividing the cells, and that goes on in a very well regulated manner. But that regulation is lost in the tumor. In the cancer cells, well regulated cell cycle is lost. Second, the cells have gone back to mimic the embryonic condition. And if they mimic the embryonic condition, they mimic the embryonic state where they will be capable of growing uncontrollably. An uncontrollable growth gives them the ability of growing into a large mass of tissue which is undifferentiated. Therefore, one can easily identify in a normal tissue when the tumor grows, those cells will not have any characteristic of that normal tissue. That is why they say that there is a growth of tumor. It is grown beyond the size of the normal organ. It is grown beyond the proportion. It doesn't have control. So it looks abnormal growth, outgrowth in that tissue, and the cells do not resemble the parent tissue. Since they don't resemble the parent tissue, they are mimicking the embryonic condition. Therefore, those embryonic genes which have been switched on in the tumor give the ability for the tumor to behave like embryonic cells and grow continuously. And such antigens are tumor associated antigens, which are fetal antigens. They are also categorized into 3 or 4 different types, which we will be discussing separately. Now we will take up the first part, that is tumor specific transplantation antigens. They are classified actually into 6 different types. And I already mentioned that the tumor specific transplant antigens are specific to the tumors and grow only in cancer cells due to abnormal mutation of some genes. And they give extra advantage for the cancer cells to grow. And they are recognized by the T lymphocytes as foreign antigens. Why? Because they are not expressed by the normal cells. They are not there in the normal cells. They are expressed for various reasons. And you know that there are cancers that occur in individual because of some bad habits also. Some people have the habit of drinking alcohol plus smoking. Alcohol and smoking go together hand in hand in such a way that both facilitate the development of cancer very easily. And there are people who smoke but do not drink alcohol. And they are habitual smokers. Large number of cigarettes they consume per day, or consume tobacco in the form of chewing tobacco, like gutka, or in the form of raw tobacco with the betel leaf, all these things. And it causes the oral cancer, or gastric cancer, or stomach cancer, or even intestinal cancer, or esophageal cancer, because the tobacco which contains the carcinogens will be bathing your GI tract continuously. So chemical carcinogenesis is one mechanism by which the tumors develop, cancers develop. And chemicals which are there in the environment consume without the knowledge, which are there in the food, which can be there in the agricultural products by accident or carcinogens. And all carcinogens are mutagens, but all mutagens are not carcinogens. In certain genes, please remember that. But carcinogens, which are known as cancer, which are not present in our body but come into our body or come intact with our body due to occupational exposure or due to habit are carcinogens, and those carcinogens are mutagens. They bring about mutation in certain genes, and those mutations are responsible for the development of cancer. Okay, so these genes that undergo mutation through chemical carcinogenesis mutagenesis process responsible for development of tumor specific transplantation antigens, TSTAs. Second example is possible that sometimes we suffer from certain viral infections, and these viruses are known to carry some homologous sequences in their genome, and those homologous sequences in their genome resemble certain sequences in our DNA. But the viral genome will have few nitrogenous bases different. And there are DNA viruses, there are RNA viruses known to cause cancers in the human beings. We will come to that example in this TSTA little later. But keep in mind, the mutagenesis could happen due to chemical carcinogenesis. The mutagenesis may happen due to viral infection, and a prolonged viral infection results in incorporation of that viral genome into the genome of the host, and that genome of the virus, which has the homologous sequence, shows recombination with our genome and replaces the correct gene by the viral gene. And the viral gene is replaces the correct gene in our cell that produces the viral protein, and that viral protein is carcinogenic, and it is responsible for the development of tumor, and that is going to be presented on class 1 MHC, and therefore it is recognized by the CTLs and NK cells. So there are viral mediated cancers, chemical mediated cancers. Then third type of cancers are spontaneous mutations, and those spontaneous mutations which happen when the cell is undergoing replication due to wrong nitrogenous base being substituted. It is either due to insertional mutagenesis, or due to deletion of some nitrogenous base, or due to translocation of one chromosomal segment to the another chromosome. Such mutations may happen spontaneously, may cause cancer. Then you'll get a new type of protein being produced due to loss of certain part of the genome due to deletion. A new protein may be produced which is not there in the normal cell, and that new protein behaves like a foreign antigen, initiates the immune response. Second possibility, a viral genome being incorporated or a new sequence of nitrogenous bases being incorporated due to insertional mutagenesis results in new protein product that may also cause cancer. Or a chromosomal segment getting translocated from one chromosome to the another chromosome that also results in different gene sequences may cause the production of new protein that also causes the cancer. So different mechanisms may be followed by the tumor, but all these spontaneous or newly expressed antigens are recognized by the sites. Okay, so the first category, the tumor specific transplantation antigens induced in rodents, and same type of tumors do happen in human beings also. If they can happen in rodents, means mice and rat, they can also happen in human beings by the exposure to chemical carcinogen, or due to constant exposure of the body to radiation. The person may be living in the environment where background radiation is very high, or maybe living near the uranium mine, or he may be working in a reactor facility, which is a nuclear reactor facility, and there is a possibility that his genome becomes vulnerable for mutagenesis. Chemical carcinogens, viral or radiation are the ones which cause mutations, and these are tumor specific transplantation antigens. Then there is a possibility that these mutated cells which have developed into tumors can further undergo mutagenesis and develop some other different antigens, and that is called a second insult. First insult of the cells results in first type of mutation, developing into tumor abnormal growth. The second insult to those cells by another mutagen also results in expression of new antigens, and those new antigens will be different from the antigens which were already expressed, and that may also initiate the immune response, and they are also type of tumor specific antigens. They are called as tumor antigens expressed on in vitro mutagenized tumor cells. Now you can do that experiment in vitro also and prove. Okay. Then the tumor antigens encoded by normal silent cellular genes. They are very, very normal genes, silent in the cell, but suddenly they become overexpressed due to spontaneous mutation. And those also may cause the expression of new antigens on the tumors. Then tumor antigens encoded by the oncogenes or tumor suppressor genes. All of us carry oncogenes in us. And those oncogenes are called as proto-oncogenes. And those proto-oncogenes perform all normal functions in our body. But due to strange incidents, those proto-oncogenes can become oncogenes and bring about transformation of cells. Similarly, all of us carry about more than 22 tumor suppressor genes in our body. In our cells, there are tumor suppressor genes which prevent the tumor from growing. But the tumor generally sees to it that these tumor suppressor genes are also mutated and abnormal proteins are produced from these tumor suppressor genes. So that is the fourth category of the antigens. Then the fifth category is tumor antigens encoded by oncogenic viruses, that is DNA viruses and RNA viruses, and the tumor antigens resulting from spontaneous mutations in the cellular regulatory genes. There are housekeeping genes in the cells which perform certain housekeeping function, normal functions of the cells, and those housekeeping genes can undergo mutation. Similarly, cell cycle regulatory genes are there. These are high regulation and strict control may lose control and develop into tumors. So six different category of tumor specific antigens could induce the immune response, and will take one by one each of them. Okay, so the tumor specific antigen expressed by tumors due to chemical carcinogen or virally induced tumors. Now let us presume that there is a carcinogen called as methylcholanthrin, which is listed at the bottom of this slide. Methylcholanthrin. Similarly, there is a sister carcinogen called as dimethylbenzanthracene. Dimethylbenzanthracene. In short form, it is called as DMBA. Dimethylbenzanthracene. Methylcholanthrin, dimethylbenzanthracene, that is DMBA, are always present in cigarette smoke. And both of them are known to be potent mutagens. To the normal cells in the tissue culture, you add this mutagen, a small quantity of this mutagen, those normal cells undergo mutation and become abnormal. If that is the rate of mutagenesis by these chemical carcinogens, imagine what happens in the cigarette smokers who are taking this carcinogen in a regular basis by smoking. Similarly, these carcinogens are also present in coal tar, which is used for, coal tar is nothing but bitumen, and bitumen is used for surfacing of the road. Okay, and coal tar, people who are working in coal mines, who work in coal mines are more prone to develop the cancers from these because they are present in the coal tar also. Okay, and people working in industries using the coal, like the power generation industry, our UPCL, which is there in Padubidri, which uses the coal for generation of power. And there, this methylcholanthrin as well as dimethylbenzanthrene will be in the atmosphere because the coal is burnt. And there is possibility that whoever is vulnerable, who inhales this carcinogen, may develop mutation in the cells and may develop tumor. Okay. Now coming to the spontaneous development of tumors because of the mutation by these carcinogens. Let us presume that you had injected this carcinogen into a mice, or you had given this carcinogen to the mice orally, or you can smear this carcinogen on the skin of the mice. The skin can be shaved, hair can be removed, and over the blank skin area, you smear this carcinogen on a regular basis for a period of about a month. Then you will see after some time the skin cancer develops in the area where you have smeared this carcinogen. And if this happens, if the mice is consuming this carcinogen through the diet and the gastrointestinal tract is exposed to this carcinogen, one fine day the rat will develop stomach cancer or esophageal cancer or intestinal cancer. If this carcinogen is metabolized and passed on to the different parts of the body, there is a possibility it may develop into a liver cancer, or it may develop into a pancreatic cancer, or it may develop into a sarcoma, that is muscle cancer, like that. So different types of cancers are possible. Even breast cancer is also possible by these carcinogens. So let us presume that in a mice, the carcinogen succeeds in development of the tumor, and two different areas in the body the tumors develop in the same mice. When the cancer develops in two different parts of the body, and you take out the tumors and analyze molecular analysis of the tumor, the strange feature will be, even though the same mice has developed the cancer of the liver and as well as the cancer of the stomach because of the same carcinogen, when you take these tumors out and observe and do the molecular analysis, the liver tumor that is developed because of the methylcholanthrin will express different sets of antigens, and the stomach cancer will express different sets of antigens. Tumor is tumor, both are cancers, but the antigens expressed by two different cancers in the same mice will be different. Let us take the cancer which occurs with the same organ, muscle cancer, sarcoma. And sarcoma may appear in different regions of the body. And you sample these sarcomas from different regions of the body, it will be surprising to note that the sarcoma of one region will express some different antigens, and sarcoma of the another region will express another type of antigen. How do you differentiate that they have expressed different antigens? It is by simple experiment that you can take out these cancer cells in different regions of the body, homogenize them, and inject them into the mice. The killed preparation of these cells can be injected into the new mice, which are genetically identical. They are called as syngeneic mice. Now the cancer developed in one group of mice. Another group is normal because it was not provided with any carcinogen. Now the developed cancer cells, you homogenize them. Now when you homogenize, the cancer cells are completely killed. There is nothing now. It is only the protein and homogeneity of the cells you inject into a particular mice. And that mice will identify all the antigens expressed by the cancer cell and produce the antibodies against that cancer cell. Plus, it will develop the CTL response as well as NK cell response. Okay, and then when that mice which is being in fact injected, which has developed the antibody, you transplant the same tumor which are used for homogenization and injected into that mice now. When you transplant the tumor into that mice, the tumor is going to be immediately rejected. Originally, the tumor was growing in the mice. And these are syngeneic mice, genetically identical. It allowed the tumor to grow. Now, you homogenize the tumor and inject it into another mice, immunize that mice developed immune response against the tumor. To that mice, you transplant the tumor. That mice reject the tumor. Reason being, it has developed the immune response against it. But the previous mice, which had developed the tumor, even though it had the antigen, it was not rejecting that tumor. It was allowing that tumor to grow. But if you had not stimulated the other mice by injecting the killed preparation of tumor cells and transplant the tumor directly to that mice would have accepted it. Okay, it would have accepted it. But now that you transplanted after immunizing that mice, means you are given the preparation of tumor in a homogeneous manner into that mice, triggered the immune response. After that, you transplanted the tumor, that mice will reject it. And those are called as rejection antigens, transplant rejection antigens, tumor transplant rejection antigens. And that proves that tumor cells are capable of inducing the immune response. And those strange antigens expressed by the cancer cells are mutagenic antigens. They are mutated antigens which are generally not seen in normal cells. Therefore, immune response builds against them. Therefore, they are rejected on transplantation. But in the first transplant, without prior stimulation of the mice, if you transplant the tumor, I've already told you that the tumor will be easily accepted. Okay, easily accepted. And it is observed that when you take the tumors from different regions of the body, each tumor which is taken from different regions of the body may express different antigens. There is no thumb rule that the antigens expressed in different regions of the body in the same animal should express the same antigens. They could be different, which can be proved only through the immune response, where in the mice you are given tumor 1 as an injection, it developed the immune response against it, and when you transplanted the tumor 1 into that mice, it rejected it. Now the tumor 2, if you transplant or inject into that mice will accept it. Why? Because it has not developed the immune response against that. It has not developed the immune response against that. So it will be considered as new and tumor is accepted by the mice. So such is the case with tumor transplantation antigens, tumor specific antigens, which trigger the T cell response. So the second category, that now you have developed the tumor in mice, let us say, by smearing methylcholanthrin on the skin. Skin cancer has taken place in the mice, and that skin cancer, you are transplanting the tumor cell as skin cancer into another mice, syngeneic mice. And when you transplant that tumor cells onto the other mice, that skin graft, the skin graft is accepted. I told you that. Okay, it is accepted because tumor has the ability to suppress the immune response. Okay, and we'll talk about those molecular details later part. But keep in mind, tumor can be easily transplanted. It is transplanted, tumor is accepted. Now let us say those tumors that are growing on the skin of the mice, that skin tumor cells, you subject them to mutagenesis in vitro. In vitro mutagenized tumor cells. Now, in vitro mutagenesis, how do you bring it? By treating them with the methylcolanthrin. Okay, first you smeared the colanthine on the skin surface of the mice, developed the tumor, and the skin cancer developed, and that skin cancer can be easily transplanted, and those cells which you have now can be grown in tissue culture. And one special ability of the tumor cells is they can be grown in tissue culture for n number of years. Please remember, you might have heard about the cancer cells which are used for the cancer molecular biology studies, and they are available in all cancer research labs. HeLa cells, H-E-L-A, H-E-L-A, HeLa cells, Henrietta Lack. And these cells are obtained from the lady Henrietta Lack, who had suffered from cervix cancer. And these cervix cancer cells were obtained from her, and they were cultured in the tissue culture. And this is a long history, and this was in the 1950s. Remember that. Till today, those cells are alive in the lab, and they are available all over the world in all cancer research laboratories. And that is the special ability of cancer cells. Cancer cells can be easily grown in laboratory condition by providing them the growth media. That's it, normal growth media. But you try to grow normal cells in tissue culture, normal cells. I'm talking about normal cells. Let me say you take out some WBCs from a normal person and put them in the tissue culture media and grow those WBCs. They will grow only for 48 hours. After that, they will transform themselves into fibroblasts and remain static. There will be no growth, and they may die also. Normal cells cannot be grown in laboratory conditions. They have to have the immortality gene of the cancer gene, that is tumor gene, for growth in the laboratory condition. So that is why I'm talking about those skin cells, tumor cells which you obtained can be grown in the laboratory condition. They are called as tumor plus cells. These are tumor plus cells. They can be further treated with a chemical carcinogen and bring about mutagenesis once again. And when you bring about mutagenesis of these tumor plus cells, which are already grown through mutation by the first smearing of methylcolanthrin on the skin, on second treatment with the methylcolanthrin, they will express second set of genes, which are going to be once again strange genes. And we'll talk about that in detail in the next class. Thank you.